Yes friends, Professor Kumar at Galaxy Tutorials, and today I bring to you a lesson from Science-2, Maharashtra State Board, standard 10th. Chapter is called Heredity and Evolution, that is chapter number 1. We have taken special efforts and put these bigger diagrams for you so that you can understand the chapter better. Keep pace with me. If you don't follow anything, keep the textbook with you, keep a notebook and keep a pen. Okay? Please make a mark what you do not understand. This chapter is very technical in our book. What do I mean by the word technical? They have just put the facts. They have not explained the facts. We can explain all the facts. I can explain all the facts. We do not have that much time because we have to finish the portion for 10th standard. But if it does not interest you, you feel something is missing, please ask. Each and every explanation can be given. In the book, this is like a history chapter. The scientist and the year are mentioned. Okay? If you want to know how we got it, where we got it, you can ask me for further details. I will begin. The chapter says heredity and evolution. And the textbook says that we have to study heredity and heredity changes. This is page number 1 of your textbook. We have to study in that transcription, translation and translocation. Then second subtopic is evolution. In this I have to teach you evidences of evolution. What is the proof that we have come from monkeys and life began so many millions of years ago. Then topic number 3. We have to teach Darwin's theory of natural selection. Darwin was the person who gave scientific explanation about how so many different types of lives have been formed from the earlier lives. We will deal with Lamarckism. Lamarck was a person before Darwin and he gave some simple theories like local stories, folk tales, Danta Katha, and we do not like today in science. Speciation is the last subtopic I will teach you. How do different species form? My friend, you know what are species. We will see species. Now, when I show you this photo, and you are not in KG or first standard, you will see that this is a cat. And you will see, this is, you will say, a fox. Why this is not a cat? Why this is not a fox? I don't know. Each and every of these pictures are different species. Chickus are not same as mangoes. You will never tell me that they are same. So what is not same? They are different species. Okay? These are not one type of dog, another type of a dog. These are different types of animal. They are different species. So this is what I will be teaching you. And the last subtopic I will be dealing in speciation is human evolution. How has man reached this stage? What are the stages before us? You have studied in 8th standard, lower classes. Man was living in the forest. Man was hunting with stone tools. Man was hunting with metal tools. We will see those stages. From monkey to man, how man is dead. So let's go into the chapter. The chapter says heredity and heredity changes. What are hereditary changes? Okay. First question people ask, tell you, remark when you go to your friends, family friends, or some function in the family. He looks like his grandfather. Her hair is like her grandmother. The nose is like father. He is like his father. So why do we compare? Because we are of the same species. And here we see heredity. One mark, define heredity. Heredity is the transfer. Father gives to his child. Grandfather gave to your father. Biological characters. They have given you all the characters you see on your body. Biological characters. From one generation to another. Okay, I give my friend gift. That is not heredity. From one generation to another. From father to son. Father to daughter. Father and mother, of course. And via genes. Don't have to give them a gift pack. They go via DNA, by genes. We will be looking at details. So 1 mark, define heredity. Heredity is the transfer of biological characters from one generation to another via genes. Okay? You must memorize the definitions as far as possible. I do not suggest you to translate, change the definition, write in your own words, etc. Of course, if the order of the words get mixed, but the sense is there, it will help you. Then, now we come to some scientists who have worked in this field. Since we are dealing with genes not what you are wearing, the DNA wala genes, this is called, study is called genetics. Okay, let us start. So book write scientist, fill in the blank, MCQ, 1 mark. First, Johann Gregor Mendel is pioneer or father, the first person who studied of modern genetics. So, 10th standard, Maharashtra State Board, okay, Science-2, Chapter 1, Heredity and Evolution. Gregor Mendel is the father of modern genetics. He started studying how this biological character. Before that, God is giving, nature is giving, we are lucky. That is not science. Okay? He gave proper explanation. This is, okay, 1886, match the column, etc. Then second, in 1901, Hugo de Vries gave the mutational theory. Here is 1901. What is mutation? Mutation is sudden change in the DNA, in the genes. That is why no two people are alike. That is why new species can be formed. We need major changes. So small changes. We go on gathering. Monkey did not become man in 1 year, 2 year, 10 year. Millions of years stories there. Okay, of evolution of life. So mutations are the sudden changes occurring in the DNA or in your genes. Who explained this? Hugo de Vries. How? If you have problems, ask me. You can access the internet and see. He worked on a 4 o'clock plant. Color of the plant changed, the flowers changed. So he realized there are mutations. We do not need to know what is not given in the book. Then third, Walter and Sutton are credited with the discovery of chromosomes. 1902. They studied chromosomes in grasshopper. They took cells of grasshopper, they had big cells, and they put under a microscope and saw, and they first saw chromosome. Oh, this is carrying DNA. Okay, genes you can't see, right? They are very small. Okay, so we see only chromosome. We cannot see molecule, atoms, etc. under microscope. 4. Then 3 people, Ostwald Avery, Macklin McCarthy, and Colin MacLeod. Okay, 3 people. They proved that DNA is present in all organisms. Of course, we will add except viruses. Now COVID virus has come. Viruses may have DNA or RNA and we don't take them as pure living material. They are on the borderline of living and non-living. We can crystallize the virus. We can keep the virus in a bottle. You can keep it for 200 years and then put it on the cell. It will still work. Okay? So that's why except viruses. Now these people worked in 1944. See the years I am taking, 1886, 1901, 44. It has been a very slow study. As the tools developed, we got better and better staining, microscopy, analysis, study of science, books. Now see how much difference it is making. Okay? And in 61, Francis Jacob and Jacques Monod. Okay, the book writes Monad, Monod, because French can't, they have got graphs and all. I'll put Monad because textbook says so. Okay, these two people gave a model for protein synthesis. How do you read the gene? How does DNA decide, control and manage protein synthesis? How does DNA work? These are the first people who gave a model. They got a Nobel Prize for that. Okay. Then we had a lot of this. We had after this recombinant DNA technique. By the way, sorry, 1961. Recombinant DNA technique has developed, which you are seeing every day. You used to see every day in the CID or Miami CSS. You see these programs, they show you that laboratory is there and we are putting the gene, we are making copies, we are making insulin today, forensics. Okay, everything is not there, darwaja tod do. We have to study biology behind that. The biology recombinant DNA technique has helped tremendously man in making so many agricultural types. Today we have no shortage of food. It is making so many medical uses, therapeutic uses. You got insulin, hormones, injections you get from the market. Thanks to recombinant. We have even mapped the DNA of humans, human genome. So recombinant DNA helps us. Okay. Now we have to learn for theory 1 mark answer. What is the use of heredity? Use of heredity science. Why am I studying this? What are the applications of heredity? Number 1, diagnosis. Best example, you are seeing COVID testing. Okay. Number 2, we are having treatment. Again, you are hearing we are going to make a vaccine for COVID now. Okay, and we have already made that remdesivir antibiotic which is available in the market, and prevention of heredity disorder. So, if somebody in my family has got hereditary problems because of DNA mistakes, okay, I should be able to cure it. Suppose blood does not clot, he has got loss of vision because of gene problem, we will be able to replace the gene. Gene therapy is coming. And production of hybrid varieties. Of both plants and animals. In the nightstand, my friends, you saw New Jersey cow is now being crossed with Indian cows, and now we are getting 22 liters of milk. An Indian cow was only giving us 6 to 7 liters of milk. Now you have read all that Sahiwal, goat, buffaloes, full charts and photos were there in your book. At that time you were just enjoying looking at the animals. But these animals have done agricultural revolution, green revolution, white revolution or the dairy revolution. Amul, as you see, milk is available, milk products are available, there is no shortage. So we are getting all. And plants, you have crossed the wheat, now wheat grows fantastic. On one plant of wheat, I am getting 200 grains of wheat. So now we are having less shortage. Rice, everything. When you go to DMart, go to a shop, you see how much grains are available. So hybrid varieties. We have done genetics, gone best and best and best, and we are trying to solve our food problems. Okay, and lastly, industrial processes. My friends, you have never realized when you go to the market and you buy Amul Masti Dahi or Chitale Dahi, you are picking it from the store, it's packed in a can. In my house, second day if I see the dahi, I open the dahi, it starts losing water and becomes sour. I keep it in the fridge, but the taste is not same to same. But this dahi, that fridge is not so cool. It has come in a truck, transportation, shaking, everybody move, you picked up, you put in the basket, he built, you put it back. Nothing happened. You see? They have got a bacteria now which is making dahi in that form with less water and dahi is staying. So, see, you are getting packed idli dosa mix. Go make at home. The dough, atta is available. You see? Insulin, when it was made from animals, first we did not know. We used to, when animals were killed, we used to cut them up and remove the pancreas and from that make insulin. Insulin was very expensive because you have to kill an animal to get insulin. Number two, that insulin also had blood, some tissue of that animal. So when I put it in your blood, reaction used to happen. People used to die sometimes. Now, everybody, home only they are putting insulin. How much insulin? 5 rupees a vial. Made by bacteria now. You see? So recombinant in industrial process, fantastic. Because we know the genes responsible. We can put that in bacterial genes and make it. Bacteria does not want insulin. We want the insulin. So, this is where, again I remind, diagnosis, we must find out what proper disease we are having. We must treat the disease. We must prevent disorders so that the child does not get the problems. Somebody has split. You will see a crack in the lips. Okay? Sometimes the palate is cracked. So, we don't want like that. We want cure for that. Somebody has blood cancer. Any type of disease you may have from genes. We produce hybrid varieties, animals, plants who are giving better and better yield to us. Okay? Pineapple, so sweet. Watermelon, small but sweet. You see, all this is apple. One month they are keeping on a store. It's not getting spoiled. It's not getting black. You see. So, hybrid varieties are very important in today's life with increasing population. And industrial processes. We make all the medical, chemical, flavors, okay, using organisms whose heredity I know. Otherwise, just can't take anything and make anything. It will never come same second time otherwise. Okay, so I must know what I am dealing with. So this is my first part in the book, heredity and hereditary changes. I will move to the second paragraph, which I will refer to my chart here. We have put up this colorful chart here, so we are going to refer to the chart. So please see the scientists, please see what they have done, which year they have done, and what is the use of heredity, and define heredity. I will remove this. Coming to the second part, okay, transcription. They are written here in yellow. Translation and translocation. What are these new names, my friend? Before going in this name, I must tell you one term, one name, which combines all these names. That is called central dogma. Dogma means a fixed rule, which cannot change. Okay? And central means everything revolves around it. Sun is center to the solar system. What is the central dogma? In all living organism, DNA is copied into RNA, and DNA is in nucleus, RNA is brought into cytoplasm, and brought into DNA never comes outside the nucleus. If it gets spoiled, gone. Originals can never be spoiled. We make a copy. Making a copy is called transcription. So transcription is the process of copying a strand, one part of it. Remember, DNA double stranded. Any one strand which you copy. So it is a process of copying a strand of DNA into RNA. How is DNA copied into RNA? The strand of DNA is there. You will see my diagram. DNA is there. A, T, G, C. This is my DNA. Okay, it will open but in the nucleus. I will assemble opposite. A, T, A, G, C. Complementary binding which you have learned in the 8th and 9th standard. Instead of thymine, I have uracil in RNA. So the body knows what is DNA and what is RNA. The other bases are same. So thymine is replaced by uracil. Thymine is present in DNA and uracil is present, as you can see, in RNA. So body can make out which is DNA and which is RNA. It will not destroy the DNA. Work is done, it will digest the RNA. We don't go on making the same protein. I don't keep on making tears all day. Okay? So we have to change. So we remove the template, the copy. So I will kill the RNA. I don't touch the DNA. This process, and then I will separate this, and then I will bring it out in the cytoplasm. In the cytoplasm, I am here. Okay? This was in the nucleus. I have removed it out. In the cytoplasm, it is translated. That is called translation. That means every 3 A, T, G, C, C, any order I am writing, and instead of T, of course, there will be U, and G, anything like this. 3 will be read at one time. That is triplet. Codon. Codon. 3 I will read from this 3. Not next 3 necessary. You can read 3, you can read 3, or you can read like this 3. You see? But depends where is the start written. Wherever he has put a start, you have to start reading from there. Read 3, 3, 3, 3, and you will get an answer. If I start at some other position, I will get 3, 3, 3, which are not same. So, from the same words, I can make so many songs. This is what you are translating into amino acids. So, AUG, for example, is amino acid 1. Okay, your GCC is amino acid 2. For example, you go on making. We have 20 amino acids which we make and 4 which we have to eat, which we can't make. So, these are total amino acids we are making. This making of amino acids by reading RNA is called translation. And amino acids, when they join, they form protein chain. So, I will have amino acid 1, amino acid 2, amino acid 3, and so on. This is a protein chain which is called, in simple language, peptide. Peptide is a chain. Many such chains I will take, twist, join, and form a protein. And you will give a name to that protein, hemoglobin. So this is how we have central dogma. DNA in your cell is transcribed into RNA in the nucleus. RNA comes out in the cytoplasm and is made into proteins. We need to keep repairing, we need to keep growing, we need to reproduce, we will need copies. And this is how copies are made from the original. And first transcription, then translation, and when it is translated, proteins are made by the process called translocate. Bring amino acid, bring another amino acid, bring another amino acid. So transcription, translation and translocation occur simultaneously. If I translocate, I translate. So transcription, making a copy of DNA, translation, okay, we are making it into codons, and translocation, we are putting in amino acid as per the order of codon to make proteins. So this is our second part in the textbook, and I will show you in the book. This is the, you can see this is like a nucleus, and there is a you can see a DNA strand. I have opened 2 strands. I want to read this strand. See, I am reading it. Okay, and instead of thymine, I am putting uracil because this is the RNA red one, blue one is DNA. So I am reading it and I am getting a copy of RNA. This copy of RNA is called messenger RNA. It is carrying the message. It comes out of the nucleus through the nuclear pore into the cytoplasm. And then 3333 make codon as you are seeing here. So there will be first a start codon. Start. You can't start wherever you like. For particular protein, particular start. And more reading 3333. So I have messenger RNA. I have ribosome. You have studied ribosome is the factory for protein synthesis. And I have tRNA, transfer RNA, which transfers amino acid as per the codon. Okay, and it brings and puts them there. And amino acids are joined. Methionine, phenylalanine, you don't need to know names. And this is how amino acids are joined to each other like bogies, and a complete train is formed, which we will call like peptide apart. And many such trains together will form the train system, which we call a protein. So this is how it is occurring. With the help of RNA, genes present in the DNA participate in the function of cell and control structure and functioning of the body. The process of RNA synthesis from DNA in the nucleus of a cell is called transcription. Okay, so we'll have to define even transcription, and this will come in the exam. 1 mark. Please write what is transcription. Okay, see, and if the code is here, some like ATG, whatever, opposite anticodon, north and south pole attract each other. You will have the opposite. So only the opposite will sit on that. And exact amino acid it can carry and bring. So now I have a luxury train, Rajdhani Express. I can't put a local train bogie into that. In a local train, I can't put a Rajdhani train box into it. So these are sorted. Anticodon, match like a magnet. And then only that amino acid will sit there. And this transport will go away, bogie will be attached. But for every bogie, engine will have to bring it there. If I am bringing bogie by bogie, engine can't remain there. Then only a bogie will join to a bogie. So this is how amino acids are joined to each other. We will see it in details. Okay. So this is where we will stop today.